So, hello everyone. We are starting today module 3, that is enzyme kinetics. So, in the last module we discussed stoichiometry and mass balance that enables us to calculate the unknown parameter in any bioprocess, because it is difficult to estimate or difficult to calculate all the parameters. So, with the help of stoichiometric relations we should be able to calculate those unknown streams. Now the question comes how to do stoichiometry balance. So we discussed, depending on unknown we need to have those many number of equations, and how to get those equations through the carbon balance, nitrogen balance, oxygen balance and hydrogen balance. In addition, one additional equation was respiratory equation. But later on we discuss it is difficult to measure change in the concentration of water. So what we discuss, based on the electron balance also one should be able to determine the stoichiometric coefficient. So we did the electron balance along with the yield coefficient, we are able to calculate the stoichiometric coefficient. Further we discuss the energetics and energy balance part. So similar to this mass balance, one should be able to perform the energy balance. So we have seen this all the living system being an open system, how the energy capture efficiency is close to 30 to 40%, so rest is lost to the environment. So as we know, in bioprocess there are two biological agents, they may be cells or enzymes. So in this module we are basically going to focus on enzymes. So in this lecture we will be discussing the basics of enzymes, their classification and nomenclature, then how enzyme works as a catalyst, followed by how it lowers the activation energy and how we should be able to calculate it. So basically this is a kind of a revision of your biochemistry. But it is important to go ahead. So first we will quickly revisit these topics. So as we know why we are discussing enzyme catalyst, because if you see all the metabolic pathways, they are all biochemical reactions, and biochemical reaction I mean this reactant is converted to product in presence of enzymes. So all are catalyzed by the enzymes. A cell can select which reactant will be combined and which can be decomposed. So what it says, depending on the activity of a specific enzyme, they are able to perform different functions. And how does it happens in living system? Because enzymes work as catalysts, and basically if you see what are enzymes, so the enzymes are globular proteins. So considering the importance and significance of enzymes, various enzymes has been produced in vitro conditions for a wide spectrum of application. Some of them I will be briefly discussing in this lecture. So as we know, enzymes, we have already discussed, it is a biological catalyst that speed up the chemical reactions in living organism without compromising the process, and in addition they are not involved in the process, they do not participate in the biological reaction. So as I told, most of the enzymes are protein, but there are some RNA molecules like ribozymes, they also act as enzymes. So, what are the features, why they are so important? Because their specificity, they are specific to a particular substrate. They do not participate in the reactions, so they are not permanently changed, and it is always possible to reuse them. And these enzymes are active at mild environmental conditions. So, it is mostly ambient temperature, and it is also important they are specific to a certain temperature and pH, because being a proteinous in nature, they are affected by the pH, they are affected by the temperature. So, for each given enzyme, their performance is optimal at a given temperature and pH. Now, once again I will re-emphasize here, the enzymes increases the rate of reaction, but they do not affect the equilibrium. So, we can quickly see few enzymes well known: amylase, lipase and protease. Amylase works on the sugar, it converts starch into glucose. Lipase, it breaks down fat into fatty acid and glycerol. Protease, it breaks down protein into amino acids. So, depending on the a biological macromolecules, there will be different enzymes. Let us say here I have listed 3 for the carbohydrate or starch amylase, for fats lipase, and for protein proteases. So, why enzymes are so significant in biology? Because they are essential for all the metabolisms. You start with the digestion, metabolism, DNA replication, detoxification, and almost all the cellular processes, you name any, all are mediated by enzymes. Now, as I told, enzymes are proteins. So, depending on the structure, the function of proteins are affected, and same is true for the enzymes as well. So, as we know, for a protein there is a polypeptide backbone, and these backbones go for this primary, secondary, tertiary and quaternary folding, and finally it end up with 3-dimensional globular structure corresponding to the minimum value of Gibbs free energy, and this is called the native conformation. So, once it is a 3D folded protein, now it has active sites, and active sites are defined as a specific region where substrate binds. So, that is why we say enzymes are specific. So, their active site will only bind to the particular substrates. So, they are uniquely fit, only certain substrate molecules can bind to the active sites. So, it looks like it is a lock and key model or induced fit model. So, in the active site, it includes specific amino acids that are helpful to carry out the reaction. So, if you see next, I have included. So, this is one example of enzyme chymotrypsin. So, you can see in the case of chymotrypsin, substrate is binding to the active site of the enzyme, and in the active site there are key active amino acid residue. You can see the red color here, and at the active site there are mainly R groups of aspartic acid, glutamic acid, cysteine, histidine, lysozyme, methionine, serine and threonine. In addition, the end amino and carboxylic functional groups are there. So in addition, many times the performance, the functional property of enzymes are achieved in the presence of cofactor. They require some non-protein compound for their function. So if this is the case, then enzyme without those non-protein part is called apoenzyme, and when they combine with cofactors, that is called holoenzyme. These cofactors can be inorganic ions like zinc, magnesium, iron, or even it can be complex organic molecules like NAD, FAD, and coenzyme A. So what it says, you need to have these enzymes, and many times if enzymes require a cofactor, then that is called apoenzyme, and when it is added with cofactor, that is called holoenzyme. Now it's important to understand this. This relates this protein structure and function. So what it says is the function of protein is regulated by the structure of the protein, and how we can tune the structure of a protein by tuning the amino acid or primary sequence of a protein. So it's very important. If the primary sequence is changed, it means it might happen that 3D folded structure will be altogether different. They might have different active sites. So that's why you say the function of that protein will vary if there is a change in the structure of a protein. So here we can outline the enzymes. We can have simple enzymes, proteins, or complex enzymes. The complex enzyme means it will be apozyme. When we will add the cofactor, that will become the holozymes, or some ribozymes are also acts as catalyst. So as I told, apoenzyme, they require the cofactors, and these cofactors can be inorganic metal ions or organic coenzymes. So this outlines how enzyme works as biocatalyst. Now next comes, we can classify the enzymes. So it is done by following International Union of Biochemistry and Molecular Biology, IUBMB, and this is the website iubmb.qmul.ac.uk/enzymes. So here one should be able to check the database of enzymes. Now this nomenclature, it can be done based on the type of reaction being catalyzed, depending on the nature of substrate, depending on the source of enzyme, depending on regulation of enzyme, or any trivial names of enzymes. So depending on this, we should be able to name a particular enzyme. So how enzymes are written? It is written as E C A dot B dot C dot D. So A refers to the class, B refers to the subclass, C refers to the subclass, and D refers to subclass. So B and C describes the type of reaction, and D describes specificity of a particular enzyme towards a given substrate. So depending on this nomenclature EC, A, B, C, D, one should be able to distinguish different enzymes. So these are the 6 main classes. The one is oxidoreductase, 2 is transferase, 3 is hydrolase, 4 is lyase, 5 isomerase, 6 ligase. So here I have mentioned the type of reaction involved. This oxidoreductase, transferase, hydrolase, they involved in the hydrolysis reactions, lyase in the cleavage isomerase transfer of functional group, ligase in the binding reactions, or you can say condensation reactions. So these are the 6 different class of the enzymes. So the first thing is the A is the class. Now B is the subclass. So for each class, like EC1 means it is a oxidoreductase, that is the main class. Now they can have many, I have listed a few, they can have many subclasses. And the third digit again, the C, it refers again different subclasses. So like for example, if I write EC dot EC1 means it is a oxidoreductase. Again if I write 0.1 means it is alcohol group is working as hydrogen or electron donor, and if I put further 1 it means the hydrogen or electron donor is NAD or NADP. So in this way, depending on this, if there is aldehyde group, the number will be 2, there is a primary amine, number will be 4, and so on and so forth. So this list is already given. So I have given these 2 example, EC 1.1.1. So 1 means class is oxidoreductase. Second subclass is enzyme acts on alcohol group, you just see the alcohol group. Third, NAD or NAD is the acceptor, this is the electron acceptor is the NAD or NADP, and last one is this is a particular enzyme or substrate specific. So this is specific to the alcohol. So basically 1.1.1.1 is alcohol dehydrogenase. CO alcohol dehydro, this is alcohol dehydrogenase. And now similarly if I say EC 1.1.1.27, so now 27 all 3 will remain same, only 27 means D. D is specific to a particular substrate. So here substrate is the lactate. So now its systematic name will be lactate NAD oxidoreductase, or its common name is lactate dehydrogenase. In place of alcohol, it is lactate dehydrogenase. So this is the classification and nomenclature of enzymes. Now I have listed a few common enzymes like proteases, they works on hydrolysis of protein. You can see this amylases, they works on the breakdown of starch for maltose production. Amyloglucosidase, conversion of starch to sugar. Glucose oxidase, they are oxidizing the glucose to gluconic acid. So these are the list of enzymes. So this lactase, protease, catalysis are main important in the dairy industry. The cellulase, amylase, protease main important in the brewing industry, where they are applied for liquefaction and clarification to supplement the malt. So when we are using the malt means that is a starch. That starch need to be first converted to the sugar before going for the fermentation. Alcohol production, we are using amyloglucosidase, a conversion of starch to sugar, and this sugar will go for the alcohol. Similarly in the baking industry, I have listed just a few, there is a long list. So now we can see what are the characteristics of enzymes specific depending on their active sites, they are very specific to the substrate. Sometimes they may require cofactors. So in the absence of cofactor we call apoenzyme, in the presence of cofactor we refer as a holoenzymes, and they have higher turnover number. The turnover number is defined as net number of substrate molecules reacted per catalyst site per unit time, that is called turnover number. So they have higher turnover number, and as I discussed initially, enzymes increases the rate, but at the same time they don't interfere with the equilibrium. So it is not going to change your product. Now these two terms are very common. Enzyme activity, it is defined as substrate converted per unit time, and when we say enzyme unit, it says amount of enzyme required to catalyze the conversion of 1 micromole of substrate per minute under standard conditions. So one should remember the enzyme activity and enzyme unit. Now once we have discussed about the enzyme, then how it works. So it basically says, as it is very specific, the most simplest model is the lock and key model. So it says E is the enzyme, it is binding with the substrate and it is forming the enzyme substrate complex. So this is the first step, step 1, and in step 2 enzyme substrate complex is dissociated in the product plus enzyme. So we started with enzyme, and again enzyme is intact in the entire reaction. So specificity means substrate binds to active site of the enzyme, and this lock and key model you can see further here. So what we say, this is the active site and this is your substrate. So they are specifically able to bind to the active site. So dotted lines you can see they are in non-covalent interactions. So only a substrate with the correct 3D configuration of R group will fit like a key fitting into a specific lock. In fact, this also explains the enzyme stereospecificity. The enzymes will only act on a specific enantiomer or isomer of the substrate. So this part we will be further discussing when we will be discussing design of the inhibitor of the enzyme catalysis. But as of now, this enzyme catalysis works as lock and key model. Now what happens, how it is able to affect the rate of the reaction? If you just see, this is your substrate, this is your product, and this is enzyme substrate complex. So your from the glucose you are going to enzyme substrate complex, it has the higher activation energy, so rate may be slower here. Now what happens in the presence of enzyme, when enzyme is present, the activation energy, if you compare these two, it has decreased, and once the activation energy has decreased, it can quickly reach to this intermediate steps or enzyme substrate complex, and further it can dissociate to the product. So you just see there is no change in the product, starting with glucose 1 phosphate to glucose 6 phosphate, there is no change in the product, there is no change even the standard free energy change of substrate and product, that is difference is just same, 1.75 kilocalorie, 1.75 kilocalorie. The only change here is the decrease in the activation energy is the low in the presence of enzymes, and that enables us to increase the rate of reaction without compromising with the equilibrium or equilibrium constant. So how this activation energy is calculated? So we follow the Arrhenius equation. So it says the rate is dependent on the temperature. So it says K is the rate, A is the constant into E to the power minus EA is activation energy, R is gas constant, and T. So rate depends on the temperature. So if you write in the log, it will be ln K equals ln A minus EA upon RT. So if I want to calculate the EA, the one approach will be to know the value of rate and temperature. So if you can perform the experiment at two different temperature, T1 and T2, you are going to have two rates K1 and K2, as mentioned here, equation let us say A and equation B, and if you subtract this equation A minus B, so we will end up with EA upon R 1 upon T2 divided by minus 1 upon T1 equals ln K1 upon K2. So, in this way we should be able to calculate the value of activation energy. Other option is, if the value of rate and this constant A is known to us, we can directly calculate the value of activation energy. Now, I have listed two examples. In the first case, rate constant K is given to us, K is 12 mole inverse second inverse, temperature is 353 K, and pre-exponential factor A is given here, A is 20 mole inverse second inverse. So, here we can directly apply the first equation, K equals to A into e to the power minus EA upon RT. So, this will be ln K equals ln A minus EA upon RT. R is the gas constant, so value of R will be 8.314 joule per mole degree Kelvin. And if you apply this, we will end up with the value of EA, that is 1499 joule per mole. Now, in the second one, at two different temperatures you are having two different K1 values. So, you can write here K1 is 33 mole inverse second inverse, at T1 is 298, and K2 is 45 mole inverse second inverse at given value of T2 of 675. So, now we can apply the previous equation. It says EA upon R 1 upon T2 minus 1 upon T1 equals ln K1 upon K2, ln K1 upon K2. So, all the parameters are known to us. If you apply, you will get the value as EA as 1376 joule per mole. So, with this I will stop here. So, today we discussed basics of enzymes, how enzyme functions, their nomenclature and classification, how they work as a catalyst, and how they influence the rate of reaction without compromising with the equilibrium, and how one should be able to calculate the activation energy. So, with this I will stop here. Thank you so much.